So, today's lecture is going to be on immunodeficiency. So, what do we mean by immunodeficiency? So, what happens is you have situations where people or animals are immunocompromised, and that means they lack certain immune functions or immune defects. And we have actually in the course of the lectures we have discussed some of these, but what I will try and do in this lecture is to put it in context of the entire immune response as such. Okay. So, in fact, in case of immunodeficiency, one prime example is that of David Vetter, who was also known as Bubble Boy, and I think all students should be aware of this case. It is an interesting case where David Vetter was diagnosed to be immunocompromised, and from the time he was born, and it was this was a sort of experiment that was done. So, the time he was born, he led a sort of sheltered existence, and so that he would not come in contact with any sort of pathogens or even just normal general microbial flora that is usually present. So, everything that was fed to him was autoclave, was sterile. So, he did not come in contact with any microbes in that, both good as well as pathogenic. So, he was actually, his life started off in a bubble, and the bubble expanded as he became bigger. And this was the idea was that as he would become older, maybe some sort of a therapy would become available that might help him. So, what happened as a result, as he was getting older, he was not in contact with other children of his age, and doctors when they had designed this failed to realize the psychological implications of their study. So, at a later stage, when they tried to take care of his condition by giving him a bone marrow transplant, and the bone marrow transplant was from his sister who was apparently normal. But at that time bone marrow transplants were done without checking for whether there are viruses or not. It so happened that the bone marrow from his sister contained some viruses, and because he was immunocompromised, the virus took over, and ultimately he passed away. But his case became very important because a lot of publicity was generated about immunodeficiency and about the experiment that was done generated a lot of interest because it also talked about the role of scientific research and ethics and also psychological problems with or psychological issues that come up with kids who grow up in completely isolated environments. So, it is something that students must look up and read up. It tells us a great deal about immunodeficiency. So, if you are immunodeficient, then you are unable to fight just normal flora. If you are not able to do that, what happens is you come down with recurrent infections. So, one of the common problems with immunodeficiency is that people come down with recurrent infections, and this usually starts off with children, because that's when these sort of this sort of this sort of phenotype is sort of becomes expressed. So, what we will do in this lecture, we will discuss some aspects of immunodeficiency. What we will although some aspects of the innate response and the B cell response will be covered, it will be mainly focused on T cell responses. Okay. Now, with respect to immunodeficiency, some we have considered. So, for example, what would happen let us say if you did not have RAGs. Now, what are RAGs? RAGs are the enzymes that are responsible for recombination, and the recombination is for the B cell receptor as well as for the T cell receptor. So, in case there are mutations in RAGs, the phenotype is that you are unable to generate functional B cell receptor or a T cell receptor, as a result of which you are severely immunocompromised because you lack the entire lymphocyte arm or the adaptive arm of the immune response, and you can see that this would lead to major problems. There are other phenotypes also that are observed. So, in case for example, you lack MHC class 1 or MHC class 2. Now, if there is a MHC class 1 deficiency, what would happen is you would not be able to select for CD8 positive cells. So, those people would have a compromised would lack CD8 positive cells. On the other hand, if there is MHC class 2 deficiency, then it would lead into individuals that lack the CD4 response. Now, in this case, we need to be familiar with certain terms, and one of the most important terms that often comes up is that the genes that are affected are autosomal recessive. Now, what do we mean by autosomal recessive? Now, there are two types of chromosomes. One is the sex chromosomes, the X and Y, and the other is the autosomes. Now, in case of XY, if there is a particular gene that is present on the X chromosome, and in case of males, if there is a mutation in this and it gets inherited, then it will phenotype itself. However, if in case of females, you will have two X's. So, even if there is a mutation in one X, you inherit one from the mother, and let us say the other one from the father is normal, you are probably okay. It's important to realize these aspects that you need often in case of autosomes, especially genes that are present in autosomes, you would need both the mutation to be inherited from both the father and the mother in order for the phenotype to be expressed, and that is why they are autosomal recessive. So, there is a distinct difference between autosomal recessive and if they are sex linked. If they are sex linked, you will get the different frequency, whereas if in case of autosomal frequency, the chances are a lot less, because you need to inherit both bad copies, one from father and one from mother, in order for the phenotype to be expressed. And I hope this part is familiar with students, and this is just simple genetics that I am sure you have sort of learnt previously, but it is important for this class that you understand these aspects. Okay. Now, how does one diagnose immunodeficiency? As was mentioned previously, that the most common feature of immunodeficiency is recurrent infections, and it starts off when when people are young. And in some cases there are cases of cancer, for example, Kaposi sarcoma or lymphomas. Now, why is this? And this is because the immune response not only takes care of microbes or pathogens, but they also takes care of tumors that develop, and the immune system is very good at trying to control both these two. So, in case the immune system is compromised or deficient, then you will have these other features that are being expressed. So, in terms of diagnosis, what is often done is one would need to measure the complement amounts in the sera, the immunoglobulin amounts in the sera, especially this is especially true in case of B cell deficiencies, and then blood counts of B and T cell numbers, and in some cases maybe function studies can also be done if required. So, you may be in some cases you do see sufficient number of B cells, but in terms of actual antibody production they are deficient, and that would show up in terms of lower amounts of immunoglobulins in the sera, and if you activate these B cells, you would maybe find that even though they look fine, they are not actually functioning in the manner that they are supposed to do. Okay. Now, what we will discuss now are what are some of the common immunodeficiencies of the innate system, and again these are aspects that we have discussed previously, but I think again it is important to put it in this one heading of an immunodeficiency. The one important one is that of mannose binding protein or MBP. This is a common problem, or it is a common immunodeficiency in fact. Now, MBP is an acute phase protein, that means it is made by the liver in response to inflammatory conditions, and the liver makes a lot of these. It is a C type lectin. What MBP does is that it binds to carbohydrates on surface of microbes. Not only does it bind, it also activates the complement cascade. So, binding as well as activation of the complement cascade is important, and what this would do is it results in lysis of the microbes. and consequently lower amounts of MBP in serum leads to recurrent bacterial infections. MBP is a very common immunodeficiency that is found, and it is very important. It also tells you about the important role of complement proteins, or important role of proteins that activate the complement system. In case there are deficiencies in this, certain phenotypes will get expressed. And with MBP, what the way it was discovered was, if you take serum from the patient, then you are not able to lyse these microbes because you are unable to bind and activate the complement. However, if you mix it with another person who is not suffering from MBP, you are able to lyse. And so that said that there was some problem or some deficiency in the serum, and it turned out to be MBP, which was discovered much later on. The other deficiency that we will talk about is leukocyte adhesion deficiency, or LAD. Now, what happens in LAD is that there are mutations in CD18. Now, CD18 is a beta subunit and which is shared on LFA on lymphocytes, or LFA is a member of the adhesion family of proteins. It is present on lymphocytes. Now, this CD18 associates, it is the beta chain, it associates with the alpha chain of LFA and is expressed on the surface of lymphocytes. Now, CD18 is also associated or a part of the complement receptor 3, CR3, that is present on neutrophils, macrophages, eosinophils and NK cells. Now, what happens is, in the absence of CD18, the alpha subunit is unable to be expressed on the cell surface, because the alpha subunit needs the beta. They will join together and they are expressed on the cell surface. Now, what happens if you do not have CD18? If you do not have CD18, there is a defect in migration, because in the absence of proper LFA, these cells are unable to adhere to the vascular endothelium, and which is a major problem. So these are two examples that I have taken MBP and LAD to tell you about importance of different aspects of the innate system. What about the B cell system? Again, in the B cell system there are two that we will consider, but the first one is X-linked A gamma glomenia. Now, A gamma glomenia is that A gamma means lack of antibodies. Okay. The other important aspect over here is that it is X-linked. It is X-linked because the gene that is responsible for this is Bruton's kinase, and that is present on the X chromosome. Okay. So that is why it is an important aspect, and that is why initially when we discussed autosomes and sex chromosomes, this aspect came up. Now, what happens now? Bruton's tyrosine kinase is an important player in B cell maturation and also in the mast cell activation. So patients who lack or who have mutations in Bruton's tyrosine kinase, they have normal pre-B cells, but these pre-B cells are not able to mature into proper functional B cells, as a result of which you have individuals with without any functional B cells. Consequently, you will not be able to generate antibodies. So this is what is X-linked A gamma globinemia. The other one that is common variable immunodeficiency. Now, over here you have low amounts of immunoglobulins. Now, B cells are there, but they are unable to make optimal amounts of antibodies. Now, why is it that they are unable to make optimal amounts of antibodies? There are several reasons for this. Several genes have been implicated. So, unlike the X-linked, where you have a single gene that is responsible for it, in the CVI, or the common variable immunodeficiency, there are several genes that are play a role. So, for example, ICOS. Now, what is ICOS? ICOS is something that we had covered during the course, our class on co-stimulation. It is closely it is a family member, it is a CD28 family member, but ICOS comes up later. You will remember that CD28 is expressed on normal T cells, but ICOS of the family member is expressed later upon T cell activation. Now, if you have in individuals who lack ICOS, they often show improper or lower amounts of the antibodies, perhaps because it probably plays some role in controlling B cell activation, so the phenotype is expressed in that manner. The other genes that are is CD19. Now, CD19 is associated with the B cell receptor together with CD21 and CD81. So, consequently, if you have mutations in CD19 or in CD81, it results in a common variable immunodeficiency, because in this case, even though you have B cells, what is happening is you do not have proper B cell signaling, which results in this. So, the other gene that is responsible is CD20. CD20 is again expressed on B cells. CD20 is important because in some cases of myelomas or B cell lymphomas, CD20 is the target, and if you have antibodies against CD20, then you can control these myelomas. So, it perhaps plays some role, but in case you lack CD20, then it results in common variable immunodeficiency. So, these are two examples that we sort of that are picked in care to refer to the importance of B cell immunodeficiencies. Okay. By and large, you must have heard the term SCID. Now, what SCID refers to is severe combined immunodeficiency. There are several genes that are involved or that might give rise to the SCID phenotype. The most important one is RAG1, RAG2. Of course, as mentioned previously, RAG1, RAG2 you will not generate the B cell receptor or the T cell receptor, and it will lead to these sort of problems. We will discuss this a little later on. The other one is JAK3, again something that we will see. The third one is the gamma chain of common cytokine receptors, and there are several of these receptors that utilize the gamma chain. So, for example, the gamma chain is common for IL-2, IL-7, IL-9, IL-4, IL-15, IL-21. Now, this is an aspect that you must have studied while doing the classes on the cytokines and cytokine receptors. The other two are actually metabolic defects. The first one is adenosine deaminase, so ADA. The other one is purine nucleotide phosphorylase, PNP. So, we will discuss a little bit about this. So, for individuals who lack adenosine deaminase, what happens is this enzyme is responsible for converting adenosine into inosine. Now, if this enzyme is not present, you have large amounts of deoxyadenosine being generated. Now, because of accumulation of deoxyadenosine, it increases the concentrations of S-adenosyl homocysteine, and these products are actually toxic to immature lymphocytes, both T and B cells. Consequently, the lymphocytes are affected, they die, and it results in deficiency of these cells. Now, in the absence of these cells, obviously it would lead to immunodeficiency phenotype, because here you have case where you do not have B cells or T cells. The other one is remember, these are metabolic defects or defects in metabolism. The other one is purine nucleotide phosphorylase. Now, in here this PNP is important in conversion of adenosine into adenine and the ribose 5 phosphate, inosine into hypoxanthine and the ribose 5 phosphate, and guanosine into guanine, which is the base, and ribose 5 phosphate. Now, deficiency in these leads to increase DGTP. This causes toxicity to the T cells, leading to deficiency. Again, in the absence of T cells, you would have immunodeficiency, because T cells are major players. It would not only affect your CD4 responses, which are important in helping B cells, it would affect the CD8 positive response, it would affect the macrophage response, because all these cells and all these networks are actually talking to each other, and if one important partner is compromised, then it will naturally affect the others, which is what is illustrated by these examples. Okay. So, I said we would discuss a little bit about this. Now, the failure of RAGs would lead to failure to rearrange the B cell receptor or the T cell receptor, and this syndrome is known as the Omen syndrome, very important. Now, over here, now this entire recombination process also involves enzymes that are involved in DNA repair. So for example, if there are mutations in the RAG or in mutations in other proteins that are involved in the repair process, for example DNA dependent protein kinase or DNA ligase 4 or Artemis, which is an exonuclease, all these would result in a failure to rearrange B cell receptor and T cell receptor, consequently leading to a severe immunodeficiency because you would lack both T cells and B cells. In fact, in the skid mice, as there is a strain of mice known as the skid mice. In this, the mutation is actually in the DNA dependent protein kinase known as Q. So over here, because there is a mutations in these mice are immunodeficient, they lack T and B cells, which gives it the particular phenotype. Again, it illustrates the importance of both recombination and the processes involved in DNA repair, which result in this. So it is a very important aspect. The other mutations that we talked about are mutations in the gamma chain, and the gamma chain, this particular gamma chain of these receptors, they are shared. So, for example, in the IL-2, IL-4, IL-9, IL-13, these all share this gamma chain, and this in fact it turns out this is the most common form of SCID, and in this case this gamma chain is again a X-linked recessive disease. Now, along the gamma chain goes functions through a protein known as JAK3. So, if there are mutations in the gamma chain or in JAK3 which is the Janus kinase 3, you would get pretty much the same phenotype. So, therefore, JAK3 knockout mice demonstrate very few B cells, very few T cells and NK cells. So, again the lymphocytic arm is sort of affected because these are important in generation of the cells and consequent activation and differentiation of lymphocytes. Okay. The next one that we will consider are going to be defects in MHC expression. Okay. Now, there are two types of MHC. I hope your students will remember. You have MHC, the classical MHC ones can be broken down into two, MHC class 1 and class 2. You will remember that MHC class 1, you have the heavy chain, you have the beta 2 microglobulin and the peptide. So, it is a trimer which is important for proper function and cell surface expression. In case of MHC class 2, it is made of two chains, the alpha chain and the beta chain, and then the peptide. There are other differences. MHC class 1 is expressed on all nucleated cells, whereas MHC class 2 is expressed only on certain cells which are B cells, which are macrophages, or basically antigen presenting cells, Langerhans cells, so on. So, what is important is that the expression of MHC class 2 is in specialized cells, antigen presenting cells, whereas MHC class 1 expression is fairly ubiquitous in all nucleated cells. So, we will come to this aspect a little bit later. Now, what would happen if you do not express MHC class 1? Now, if you need to remember that when we discussed MHC, we had said that MHC proteins are there are three different aspects. They are polygenic, that means you have different genes that are responsible for it. They are polymorphic, okay, that means within the genes there are certain changes that make them distinct. And the third thing is they are inducible with inflammatory cytokines, namely interferon gamma would be a good example. Now, the polygenic is important because if you remember in mice you have K, D and L, in humans you have HLA A, B and C. So, if let us say there is a mutation in one, you have other MHC class 1 alleles that can be expressed. So, therefore, the chances of MHC class 1 deficiency in this respect is somewhat less. The problem comes is that the heavy chain is associated with beta 2 microglobulin. So, if there are mutations in beta 2 microglobulin for example, so then you have improper folding of the MHC class 1 and you will have improper function of MHC class 1, which will result in MHC class 1 deficiency. Now, there are other reasons also for it. Remember, MHC class 1 molecules bind to peptides in the endoplasmic reticulum. And how do they do that? Because you have transporters that transport these peptides from the cytosol into the ER. Now, if there are mutations in the transporter associated with antigen processing, you will have problems again, because in the absence of peptide, the MHC class 1 molecules are not able to fold properly and are not able to reach the cell surface for proper functional role. So, these are all problems with this. So you may result in deficiency in MHC class 1 expression in the assembly factors, namely the beta 2 microglobulin and the transporter associated with antigen processing. In fact, what was shown is nonsense mutation in TAP2 results in low expression of MHC class 1, but and there is no peptide presentation. Now, what happens if there is no MHC class 1? The most important thing is that proper selection in the thymus is not there, as a result of which you do not have the CD8 positive cells. So, if you are not having the CD8 positives, one important part of your adaptive immune response, which is the CTL part, is compromised. So, consequently, you have the phenotype is expressed later on, because you still have the CD4s and they can do something, they can help boost, and it is expressed late, and it results in recurrent severe pulmonary infections, and the disease severity with respect to survival varies with patients, and it varies because it depends on where the mutation is, how much of an effect it has, whether you have, if you have absolutely no MHC class 1 expression, no functional MHC class 1 expression, versus low, or there is low, but it is still good enough to get some sort of responses going. So, that is what it there is a variability, because it just depends on the type of mutation and what is the is the effect it finally sheds on. Now, in case of MHC class 2, what would happen is that again you would need now MHC class 2 is also polygenic. So, in the case of humans you have DP, DQ, DR, in case of mice you have IA and IE. So, therefore, mutations in either of this resulting in absence of MHC class 2 is not going to be all that likely, because let us say if DP is affected, there is DQ and DR that could still take care. But over here the problem in case of MHC class 2 often has to do with expression, and its expression because of some key transcription factors which are important in their expression in certain in these specialized tissues, and two of them are C2TA, which we had discussed, and the RFX5. So, these are transcription factors which bind to MHC class 2 and MHC class 2 family members like DM, so on, and are responsible for generation of their transcripts. So, they play important role in their transcriptional expression, and if these transcription factors are compromised, it would result in diminished MHC class 2 expression. So, with diminished class 2 expression, what would happen is selection of MHC of CD4 positives would be affected, therefore, you would have low number of CD4 positives, and but the CD8s ought to be fine, and it is often associated, this sort of is often associated with viral infections, candidiasis, which is a fungal infections, bacterial infections, and septicemia. Okay, now defects in T cell activation. Now, in this, if you have T cell activation defects, you would have presence of normal or elevated number of T cells, but they would fail to proliferate or produce cytokines in response to mitogens and antigens. So, in this case, the absence of T cells, and then the absence of proper activation are good examples. So, some of the ones are, in fact, if you have the CD3 epsilon, remember the T cell receptor is there, it is associated with the CD3 proteins, and if the CD3 epsilon is not there, then what happens is that for the T cell receptor, you need the T cell receptor and the CD3 to properly to be expressed as a multi-protein complex, and this multi-protein receptor complex then goes up to the cell surface, and if some of the components are not there, then there is improper assembly of the T cell receptor, it cannot go up to the cell surface. So this is what happens in CD3 epsilon lacking mice. You have no T cell development in the thymus. We have very few double positives and single positive T cells. Then the other example is ZAP70 deficiency. What is ZAP70? ZAP70 is zeta associated protein, and the molecular weight is 70 kDa, which is why it is it has been given the term as ZAP70. So in this case, again you have recurrent infection similar to SCID patients. You have normal or elevated numbers of CD3 positives and which are CD4, but there are no CD8 positive T cells. Now this in case in humans, in fact what is seen is there is deficiency in both CD4 and CD8. So the question then becomes is why is it that you have normal CD4 numbers in case of mouse. Now what is possible is that in the thymus maybe some other tyrosine kinase member like SYK or something could bind and it could result in selection of CD4s, whereas in humans that sort of is not the human ones are not able to be able to complement that, as a result of which you do not have you see a deficiency in both CD4 and CD8. In case of mouse there are some differences, as a result of which you will get some selection of CD4s in the absence of ZAP70. So there are these clear differences between in some cases between mouse and humans, and this is a good example of that. But nevertheless the fact is ZAP70 is a major player and it leads to this sort of deficiency. The other one, the other T cell activation defect which results in compromised individuals is going to be your mutations in your STIM1 and ORI proteins. Now you will remember that STIM and ORI are important in mobilization of calcium into T cells, and you need proper for proper T cell activation you need a spike in calcium, intracellular calcium, but the calcium amounts need to be sustained over a period of time, so that there is sufficient calcium to activate calcineurin, to activate NF80. So you can turn on the signals, and there is proper gene expression. So individuals who have mutations in STIM or in ORI, STIM is the sensor, ORI is a calcium channel which allows extracellular calcium to enter into T cells, you will have mutations in these. So something that is important for you all to think through, and also the fact is that this aspect was sort of discussed in the T cell activation class, and it is better that you familiarize yourself with this aspect. Okay. The other one that I would talk about is the Chediak-Higashi syndrome. Now the Chediak-Higashi syndrome is a very interesting cell biology aspect. Now over here there is a problem with trafficking. Now as a result of which it affects neutrophils, it affects cytotoxic T lymphocytes and NK cells. Again the phenotype is these people these are susceptible to recurrent respiratory tract and other infections, and the mutation is in LYST, which is a trafficking defect. Now because of this what happens is you have giant lysosomal granules in neutrophils and melanocytes, sometimes resulting in albinism. Albinism means melanin is degraded, as a result of which you have white patches. Abnormal and these abnormal lysosomes cannot fuse with phagosomes. These ingested microbes cannot be destroyed. There is an absence of CTL and NK killing. Remember the granules are affected because of this trafficking problem, and since the proper formation and the granules are not there, there is improper CTL and NK cell killing. And the disease can be fatal if you do not treat it with bone marrow transplant, and this is something that we will see. Immunodeficiency, the only way that you can really treat immunodeficiencies are going to be through bone marrow transplants. I mean, whether it is David Vetter where the bone marrow was failed because at that time viruses in the donor transplants could not be detected, but now they are able to detect it. So at least chances are reduced, but this is an important aspect that transplants, bone marrow transplants is unfortunately the way to go about it, and perhaps the only real practical solution. Okay, the other is the thymic development and differentiation. Now, over here we will discuss DiGeorge syndrome. Now, DiGeorge syndrome results in a reduced number of thymocytes and resulting in improper formation of the third and the fourth pharyngeal pouch during early embryogenesis. That means during development, you have improper formation of this region, which contributes to the thymus, which and other parts, including the heart. So, if so DiGeorge syndrome is because of deletions in a particular region in the chromosome, which is chromosome 22q, there are several phenotypes and observed, because this again would depend on the types of mutations that are observed, and it affects, and depending on the kind of mutations, it will affect the thymus, it will affect the parathyroid glands, and it will affect parts of the heart, which are all close by linked. Remember, the thymus sits right on top of the heart. So, the parathyroid, thymus, heart, they are all sort of linked, and they come through a common developmental thing, which is the third and the fourth pharyngeal pouch, which gives rise to these organs. So, if there is a development defect because of some mutations in chromosomal 22q, then these sort of things result in. So, the closest to DiGeorge syndrome in terms of a mouse phenotype is that of the nude mice. Now, why is it called nude? It is called nude because these mice lack hair. So, their skin lack hair, and so it gives them, that is why the name nude. So, these mice have two phenotypic defects. One is of the nude phenotype, a lack of hair. The second is the absence of a thymus. Now, in the absence of a thymus, what would happen? You would not be able to generate sufficient number of T cells. Now, and why is this? And the mutation has been found. There is a defect in a transcription factor known as FOXN1, which belongs to a family of a winged helix transcription factor. Now, nude mice are extremely useful in terms of immunology. They are extremely useful because in the absence of a good T cell response, what you could do is you could do allografts, or in some cases even xenografts. So, you can take transplant tumors, especially tumors from other strains of mice, or even human cells, if you want to study whether their chances of developing tumors, these can be transplanted onto nude mice, and then you see if these develop tumors or not. Remember, in case of tumors, the T cell response plays a very important role, and so you are able to now check or study if something that you can actually culture these cells to find out which indeed are the cells that would ultimately turn into tumors. Especially in case of human cancer studies, it becomes difficult to do these in vivo. So, a good way to study is to use nude mice. And so, these are done because, and you are able to do it because because the nude mouse, because it lacks a T cell response, will not be able to generate a good allograft or a good anti-allograft or a good anti-xenograft response. And so, people have made use of this, or scientists have made use of this to use as models for growth of tumor cells. Cytokine defects. Now, in this case, we will discuss some important cytokines. IL-2 is clearly an important one. Now, there are two types of mutations that need to be thought about. One is defective in production of IL-2. So, for some reason there is a defect in production of IL-2, and what has been found is what one would expect with the IL-2 knockout mice is that because they cannot produce IL-2, there would be some problem in generation of immune response. In fact, what was found in the IL-2 knockout mice is that you have an autoimmune like situation. You have hyperproliferation of T cells, and the T cells are primarily of Th2 type. There is a deficiency in CTL function. Now, initially where these mice develop, they are okay. As they age, they develop this autoimmune syndrome. And why is it that they are able to do it? Here we have discussed this aspect before. Is IL-2 is important for the survival and maintenance of T regulatory cells. So, with time, as T regulatory cells decrease, you have an increase in autoimmunity, because the normal suppression is that the Tregs do are not being done. Consequently, you have increase in autoimmunity. So, this is one aspect. Now, this is also seen in cases of where you lack CD25 or the IL-2 receptor alpha. Now, CD25, you may remember, is rapidly induced upon T cell activation. It is a good marker for T cell activation. And what was seen in mice that lack CD25, again young mice, they have normal T and B cells. Now, adults, you have large lymphoid organs because of polyclonal expansion of T and B cells, and they develop autoimmunity. So, the phenotype that is shown by IL-2 knockout mice and CD25 are reasonably same. Now, the same does not hold true for the IL-2 receptor gamma. Now, IL-2 receptor gamma, as was pointed out, results in severe SCID phenotype. That is because the gamma is shared by different cytokine receptors. So, just so that I mentioned this just so that students do not get too confused, and they realize the difference between CD25, which is IL-2 receptor alpha, and the IL-2 receptor gamma. You get a more severe phenotype, and that is because the gamma is shared by the other cytokine receptors. Now, in some cases, you may have situations where not only IL-2 but other cytokines are not produced, like for example IL-4, IFN gamma. Now, in this case, what has been shown is that this is deficiency in the NF80 or the nuclear factor present in activated T cells, which is induced upon T cell activation, and which is important not only in production of IL-2 but of other cytokines. In the absence of this cytokine, you have defect in production of several cytokines, which gives you a major phenotype. Okay. Then the second one that we will discuss are mutations in interferon gamma and its receptor. Now, these were uncovered especially in cases of young infants who were given BCG. Now, if infants who lack interferon gamma or interferon gamma receptor, or if they lack IL-12 or IL-12 receptor, they have the same phenotype. Now, BCG is a live attenuated vaccine. It is a good pediatric vaccine against tuberculosis, and when given, if the person is or if the infant is immunocompromised and lacks these important cytokines, it is unable to fight BCG, and there is disseminated disease. This disease is known as BCGiosis. And the children are highly susceptible to mycobacterial infections. And in fact, in mice lacking interferon gamma or its receptor show normal development of the immune system, but they are very sensitive to infections with microbes. So, for example, Listeria, Vaccinia virus, other intracellular pathogens, and so on. And what you see is there is increased CFU upon infection these pathogens, and this sort of illustrates the important role of interferon gamma receptor in and actually also IL-12 and IL-12 receptor in playing a key role in increasing host immunity to infections. Okay. Now, there are two cytokines that we had discussed, but we had not studied in greater detail, and I thought we would do that. So, in fact, both these cytokines actually do not result in what can be considered as immunodeficiency. They are like IL-2 in that respect, in the sense that there is an autoimmune phenotype that they display. But this is an aspect because we are studying the role of different cytokines. So, I thought I would put them over here. Now, TGF beta and IL-10 are the ones that I have in mind. It is very interesting because both come under the category of immunosuppressive cytokines. Both TGF beta and IL-10 would be ones that are well known to sort of down modulate immune responses. But the phenotypes of this are very distinct. TGF beta is involved in apoptosis, growth suppression, because it increases the production of the cyclin inhibitors. It regulates Tregs. In fact, TGF beta alone induces or is responsible for the generation of Tregs. Now, however, if TGF beta plus IL-6, then you will have different type of cells coming up, and those are the Th17 cells, and that is an aspect that we had studied during our class on T cell subsets. So, very interesting. TGF beta alone increases Tregs, because that is important, but if the TGF beta is given in combination or is present in combination with IL-6, then it gives it an advantage for the Th17 positive Th17 group of cells to develop. In case of a little bit about TGF beta signaling, again this is an aspect that we had not looked in or studied. Now, TGF beta dimers bind to its receptor. These receptors phosphorylate a receptor regulator SMAD, and this is known as the R-SMAD. Now, the R-SMAD goes and binds to a common, or the C-SMAD, and together this heterodimer enters the nucleus and acts as a transcription factor, and which gives rise to the different effects that it has in apoptosis, growth suppression, so on. So, that is a primary way by which TGF beta functions. The phenotype of TGF beta knockout mice is that it results in systemic autoimmune disease. As multifocal means at different places. Inflammatory autoimmune disease, you have increased circulating antibodies to nuclear antigens, deposition of immune complexes, there is uncontrolled lymphocyte activation differentiation, increased expression of cell adhesion and MHC proteins. Now, in this it turns out that the CD4 positive cells play an important role. Now, what is the evidence for that? There are two ways, that is, if you breed now the TGF beta knockout mice with the MHC class 2 deficient mice, then a lot of these inflammatory symptoms that are seen are reduced. Okay. The other way is if you inject anti-CD4, which means you deplete the CD4 population, then you lower inflammatory responses. So, this sort of tells us that this heightened inflammatory responses that are seen in TGF beta knockout are due to the uncontrolled proliferation of CD4 positive T cells. So, again TGF beta is an immunosuppressive cytokine, it sort of reduces inflammation, and this is just proved by the TGF beta knockout mice. What about IL-10? Very interesting. Now, IL-10 is produced by monocytes and some amounts are produced by lymphocytes. It is anti-inflammatory and it is suppressive. Now, in IL-10 there is less effect on systemic autoimmunity. By systemic autoimmunity means, like for example, in TGF beta you see such a huge inflammatory reaction that in fact inflammatory responses that are seen by the mice, that sort of is not seen. Such a dramatic response is not seen by the IL-10 knockout mice. In fact, what is seen is that it causes spontaneous mucosal autoimmunity. In fact, it turns out that it is a key regulator of the intestinal immune response. And why do we say that? Because the lymphocyte development and antibody responses are normal, but the mice are growth retarded and anemic. And why is it? Because they suffer from chronic enterocolitis. Colitis means inflammation of the colon. There is mucosal hyperplasia of the intestine. Hyperplasia is proliferation. There are inflammatory reactions, abnormal expression of MHC class 2 on intestinal epithelium. There is bowel inflammation due to enteric antigens, and this you can sort of reduce it if the mice are sort of bred in again in sort of bubble chambers where they are not exposed to normal flora that we sort of live in with. Okay. So, how does one treat immunodeficiency, especially in cases of B cell immunodeficiencies like X-linked A gamma globulinemia due to mutations in proton tyrosine kinase or the common variable immunodeficiency. There is patients are injected with gamma globulins, and so these gamma globulins are got from pooled blood donors, and so this is sort of helpful. Then in the other case, as I have said, in terms of immunodeficiency, the practical mode of treatment are bone marrow transplants, and bone marrow transplants under careful conditions, and these days this is possible because medical technology has improved a great deal since the days of David Vetter. So that is the bone marrow transplant is the real practical sort of solution to immunodeficiencies, which can be really life threatening, because all the time you are coming down with infections, and if it's a really if you are infected with extremely pathogenic, then it can be severely life threatening. Then there is gene therapy, but as I have said, how much gene therapy is applicable in practical terms is not clear, but bone marrow transplants is perhaps the way to go about it. So, we will summarize this lecture. So, we started off with what is immunodeficiency, and over here it is the absence of a large part of the immune response. So, for example, if you have rags, then you have obviously a major hole in your immune response, or you have smaller ones like mannose binding lectin, a key protein that is important, but nevertheless the phenotypes are depending on the mutations, the phenotypes are often the most common one are recurrent bacterial infections, and these can be diagnosed. You can diagnose, you can check your complement levels, you can check your MBP levels, you can check for antibody, what is your basal antibody. If your basal antibody levels are really low, then perhaps there is a B cell deficiency. Then you can finally actually check for numbers of T cells, B cells, neutrophils, so on, and also check for function. And the treatment, as mentioned, that the most likely treatment or the most practical solution is going to be that involving bone marrow transplants. We discussed different diseases. So, we discussed in innate, we discussed the MBP and the leukocyte adhesion deficiency. In B cells, we discussed the Bruton's tyrosine kinase one, and the other the common variable immunodeficiency. Now, common variable immunodeficiency, there are several reasons for it, which and the several genes involved, ICOS, CD81, CD20, so on. We discussed the bare lymphocyte syndrome, bare lymphocytes, because the lymphocytes there are less number of lymphocytes, the lymphocytes are bare, and that is because you lack MHC class 1 or MHC class 2. The reasons for that are ones that we discussed. In case of MHC class 1, you lack beta 2 microglobulin or the TAP transporters. In MHC class 2, the specialized expression are responsible for certain transcription factors. C2TA is really a key player in that, and you have the other one also, which plays an important role. We discussed T cell selection, in which case DiGeorge syndrome, which is actually a developmental defect, and in T cell activation we discussed ZAP70. We also discussed the Chediak-Higashi disease, which affects the trafficking defect, and so the proper granule formation or proper rhizosome formation is not there. As a result of it affects CTL and NK function. So, overall in terms of immunodeficiencies are really important, and what immunodeficiencies have done is that it has told us about the importance of the proteins that play such an important role in the immune system. Thank you.